Okay, so it's 10 o'clock. Why don't we get going? Welcome everyone to our first Gulf Coast Consortium Immunology Cluster Symposium. This one obviously focusing on COVID-19, on immunity, vaccines, and epidemiology, trying to capture the many different presentations that we have lined up, going through almost noon today. So I wanted to thank Suzanne Tomlinson and Kara Kars for really doing a great job in helping to put this non-in-person Zoom meeting together. Thank you very much for all the help. And so just a reminder to the speakers, it's kind of like speed dating. We're gonna go boom, boom, boom every 10 minutes. We're gonna try to give a warning when you're coming up on the end. And I hope I don't have to cut you off in mid-sentence. So please try to pay attention to that 10-minute limit. And let's go ahead and start. Our first speaker is Chad Creighton from Baylor College of Medicine. Chad?
Okay, great. Can people see these slides?
Yes.
Yeah.
You can see. Okay, great. We're good to go. Okay, so yeah, I do work in bioinformatics. I do a lot of RNA-seq analysis and integrative analysis and analysis of public data. Most of my work is in cancer, and we do a lot of collaborations in cancer. And so we're kind of trying to shift a little bit to applying what we've learned in cancer to looking at data on SARS-CoV-2 and transcriptional response data in human lung cells. This is all taken from public data. So the goals of this is there was this public gene expression profiling data set that was generated by the Tenover lab, and they put that up on Geo. This is human lung cells, lung cell lines that are infected with SARS-CoV-2. And so we wanted to do a preliminary analysis of these data as a starting point for how, this might be integrated into ongoing research activities into SARS-CoV-2. This is looking at the response of the host cells to viral infection. And so taking the data, we can get like a set of top genes differentially expressed. There's actually a number of cell lines and a number of treatment groups represented by these data, and they've expanded on this data set, and we're working through it. But this initial data set they made available had like two lung cell lines, NHB E and A549. A549 is cancer cell line, but they have like a mock group and then a SARS-CoV-2 infected group. So yellow on this heat map, we got genes in the rows, treatment profiles in the columns. Yellow is relatively higher expression compared to the corresponding control. So black doesn't mean zero. Black means whatever reference, whatever levels the control was. Yellow is going to be higher than that, and blue is going to be lower than that. So we can just focus on the SARS-CoV-2 groups for now. We've got like a set of genes that are up in both cell lines within, 24 hours of infection, and then genes that are down or blue in both cell lines. So we want to take that as a SARS-CoV-2 response signature and probe that for pathways and even look at other data sets. So if you look at like Go term categories, functional gene groups, several of them pop up in the genes that are more highly expressed with the SARS-CoV-2 infection. A lot of things relevant like cytokines and type 1 interferon response and immune signaling and responsive virus and so on. So a lot of relevant gene categories. We'll get pathways. We've got a number of pathways that come up, like this type 2 interferon signaling. You can diagram it out, and there's a lot of genes that are up with infection that also come up in this pathway, a significant number. Also looking at cytokines and inflammatory responses is a different pathway. I think David Corey might be particularly interested in this pathway. Others might be interested as well in terms of the cytokine storm. So a lot of players, including IL-6, IL-1A, IL-1B, CSF, CXL1 and 2. So these are coming up in a lot of places in this pathway. Now, the other thing we did was take this response signature and look at external data sets. Now, right now, we don't have a lot of data on SARS-CoV-2 that's publicly available. But from Geo, we can download other expression data sets of viral infections from maybe other viruses in patients and model systems. So we've collected a lot of public data at this point for viruses other than SARS-CoV-2, viruses including RSV, influenza, MERS, SARS-1. And so we're going to probe these external data sets with our SARS-CoV-2 transcriptional signature to see how the overall patterns may be similar or differing for the other viral infections. So here's an example data set. We've done several of these. And this is looking at SARS-1, and this is a mouse model. It's taken from mouse lung after infection. So control group, mock group is on the left here, and then we've got different, different versions of the SARS-CoV-1 infection on the right here. So we've ordered these genes based on those that are first up with SARS-CoV-2 in our other data set, and then those that are down with SARS-CoV-2. And there's a nice yellow versus blue pattern here that we see. So of the genes that are up with SARS-CoV-2, most of these are up with the SARS-CoV-1. So there's a lot of similarities in response between these two coronaviruses in terms of the host response. Other data sets, we've looked at other viruses, and so there's a couple differences here with this data set. We've looking at, PBMCs in infants that either had influenza or RSV. Again, we've ordered the genes based on those that are up with SARS-CoV-2 versus those that are down. There is a lot of yellow here for those that are up as compared to the healthy control, so that's interesting. So there's a lot of maybe commonality with influenza and RSV infection. Now, a couple differences here is that these are PBMCs. There's not lung cells, and obviously other viruses, as well as maybe platform and study differences as well. So maybe not everything's going to correlate, but a lot of things are correlating. So that's interesting. This is another data set. This is RSV infection in patients. Again, PBMCs, acute versus recovery. The genes are up with SARS-CoV-2. A lot of these are up in the acute phase, and then they go down in the recovery phase. So some similarities with SARS-CoV-2 and viruses such as RSV. This is a nice data set of mouse lung and a host of different infections in mice, some viruses as well as a couple different bacteria infections. And so we've got control on the left here and then infected on the right. And so a lot of genes that are going up, they're also up with our SARS-CoV-2 signature. A lot of genes blue that are down. Interesting about this data, it says it has both PBMCs and lung cells. So I guess PBMCs are the pink, and then the peach are the blood. And some genes are going up in the lung that you actually don't see going up in the blood. So it may depend on the cell type in terms of what the specific responses are. Some genes may not go up in the blood as they do in the lung. Now, the last thing we looked at was there's additional SARS-CoV-2 data that's been generated and from the same group, and we're looking at those as well. Not just SARS-CoV-2, but also other viral infections. So we've got all these data we can pan across the different treatment groups. But there's another cell line that's also been added. And just as a final point, we've got now CalU3 along with A549 and NHB3. A549 and CalU3 are cancer cell lines. And again, the NHBE is, bronchial epithelial cells. And so these are genes going up in any one of the cell lines. So yellow is high relative to its corresponding control within that cell line. And there's a lot of genes going up in the cancer cell line, A545 and CalU3. Not as many going up in the NHB3. We can define a set of genes that are kind of common across all three cell lines. But it does seem interesting that there are some genes that are specific to some cell lines. I don't know if there's a story there, if there's reasons why the response would differ a lot between the different cell lines, but that's something that we're thinking about as well as looking at other data. So I guess I'll just end here on kind of the summary slide. Again, we defined a transcriptional response in lung cell lines to SARS-CoV-2 infection in vitro. Look at pathways. A lot of interesting pathways come up. And then looking at across other data sets from other viral infections, there seems to be a lot of concordance in our signature pattern versus the signatures that we see in other virus infections and other systems and studies. So this data is a tool, so people that have questions on the data, we can kind of pull that out and kind of look across different data sets and different treatment groups. And I guess if people are proposing projects that need RNA-seq analysis, that's something my group can do. That can be like time and effort on grants or contract or something like that. So we could, if people are needing bioinformatics support for proposed projects, and that's something that we'd be interested in as well. So I'll stop there. Thank you for your time. Oh.
Sorry. Okay, I think I'm on again. Thanks very much, Chad. That was an excellent presentation, a huge amount of work on your part. Again, if anybody has any questions, please type them in. We have one. Chad, did you see upregulation of COVID-19 receptors, including especially ACE2?
I did not. No. Yeah, they don't go up, I guess, when it's effective. The receptor is constantly expressed, I guess, and once it gets into the cell, that's what we're seeing, the changes that are initiated in response to the viral infection. But at that point, the virus is already in, so it doesn't turn up the receptors we see.
Great, thanks, Chad. I think we're at the 10. Let's see. All right, one more question. Oops. I think, well, we better move. We're at the 10-minute mark. We better move on. Thank you very much, Chad. If any other follow-up questions, feel free to send your questions directly to Chad. But let's move on. And our next speaker is going to be Dr. Mohamed Hussein from Baylor College of Medicine. And again, just a reminder, please do type your questions in, and we'll try to get to those. But please, speakers, try to keep your talks limited to under 10 minutes. So we can get those questions in. And please take it away, Dr. Hussein.
Okay, good morning. Can you guys hear me?
Yeah. Mm-hmm.
All right. So some peculiarities regarding COVID-19. One, children seem to be the least affected in terms of severity and possibly even frequency, which is totally counterintuitive to what we know about respiratory tract infections, where kids tend to be affected with more frequency and with greater severity than adults. Two, severity of the infection and the mortality so far seems to be affecting less developed countries more than more developed countries, which is again very counterintuitive. You would expect more burden on the less developed countries with typical infectious diseases. Three, there is big discrepancy in the severity of infection among patients who have similar clinical profile. You can have two people having the same clinical profile, one dies and the other one actually doesn't have symptoms at all. Looking into the route of transmission of the virus as a possible explanation for these peculiarities hasn't been seriously considered or looked at. We know that respiratory viral infections in generally have access to the body mainly through the nasopharynx. Ocular involvement was thought to play a less important role. However, that role hasn't been properly studied. And for the past couple of 3 weeks, we learned a lot about the relationship between the eye and COVID-19. Now we know that the virus can replicate in the tears for a long period of time. We know from one big study that 30% of patients can have ocular involvement. The majority of those that have ocular involvement would end up having severe disease. And the very interesting study that came like a week ago showed that the virus had very, very high affinity to the conjunctiva and the ocular surface, very much similar to the affinity to the epithelial lining of the respiratory tract, 100 times more than the affinity of SARS-CoV-1. So the virus loves the eye, and the eye seems to be a problem with COVID-19. And not only that the eye seems to be a problem, I think it is the problem when it comes to severe disease. Ocular involvement and immunity can explain all the peculiarities that I mentioned. The fact that children are at risk for less risk for being infected and with less severe disease can be explained by the ocular immune differences between adults and kids. Adult eyes are immune privileged organ, which means that they have less immune response or suppressed immune response, which is supposed to be something protective for the very vital organ like the eye. Kids, it's not like that. It's not an immune privileged organ. They actually have the opposite. They have strong immune responses, which may have protected kids, as I will mention in a few seconds. Another observation is what I said, the less developed countries having less mortality from that disease. And again, the eyes of people living in less developed other countries are not immune privileged, or a lot of times it's not immune privileged, less immune privileged compared to the eyes of people living in developed countries because of endemic ocular infection, which can allow more innate immune response that allows the eyes to fight infections more reliably. One common endemic ocular infection shared by most of the countries that have least for the mortality is something called trachoma. If you guys don't know what that is, it's basically a service disease, a disease of the eye caused by chlamydia, and it's the commonest cause of blindness in less developed other countries. You can see here the map for endemic trachoma countries. It really matches the map of the less mortality from the disease. Interestingly, if you take Ethiopia and South Sudan, for example, they actually have the highest, they have the most endemic for trachoma, and these two countries have a handful of cases and they literally have zero mortality, Ethiopia and South Sudan. Another interesting point is that countries where wearing glasses is an epidemic, like the Asian border countries, seem to control the infection at faster rates and tend to have less mortality from the disease, taking like Singapore, South Korea, Taiwan, parts of China. Myopia, which is nearsightedness, is actually an epidemic there. Like in Singapore, it's 90% of people are myopic. In South Korea, it's like 75%. And going to Europe, the Germans have the highest prevalence of astigmatism and myopia. And we know that actually the Germans had less mortality compared to their neighboring border countries. So it could be that protective, wearing glasses lessens the severity of the disease. From this immunologic data and epidemiologic border data, it seems that the eye is a problem and can be the problem that directs the severity of the disease. If the eye gets involved because the weak and innate immune response typical for adult border eyes, the virus would replicate unchecked on the ocular border surface, tears, and then delivers large amount of virus through the tear duct system into the nasopharynx, challenging the immunity, the natural immunity, and leading to severe disease. This is not going to happen in children because they have better immune response to their eyes. It's not going to happen in less developed border country patients, some of them, or people, some of them, because their eyes are not immune privileged for the most part because of the trachoma. So as such, topical ocular medication may be an ideal treatment and maybe ideal prophylaxis for COVID-19. It would exert its effect on the eye, sterilize the eye, would drain through the tear duct system into the nasal cavity, and then there would be like rapid absorption of significant amount of it by the vascular nasal mucosa, and it would have a systemic effect. Cyclosporine is one medication that was found by a large study to have a strong antiviral effect against coronaviruses, specifically SARS-CoV-1. And this nice for the study, multiple countries in Europe shared in that study, and they found that a very low micromolar non-cytotoxic concentration of cyclosporine is capable of killing SARS-CoV-1. We know we have cyclosporine as an eye drop, which is kind of fortunate. It's a drop that is used for treatment of dry eyes. It's been used safely and effectively for years. There are two concentrations of it. The Restasis is the most popular and common for the ones. This is 0.05%. We calculated the dose on this drop to be 13-fold the lethal dose for SARS-CoV-1. We have another one that just came in the market last year, which is double that doses, can be more effective. We have both FDA approved Restasis in 2003, and the Sequa, which is now 0.09%, actually was approved last year, and they both have been used safely and effectively, no systemic or topical side effects whatsoever. So if there is evidence of the literature that cyclosporine actually can kill coronaviruses, specifically quoting about SARS-CoV-1, it's not really totally crazy idea to assume that it may have a similar or close effect on SARS-CoV-1. And since we have these drugs available, they are safe, no topical or systemic side effects, it may be warranted to look into the effectiveness of these drops in reducing the viral load in patients that have COVID-19, and more importantly, to use it as a prophylaxis. So in summary, the ocular pathway may be an important source for COVID-19. We now know that it's there, that the virus loves the eye, and it may be the directing force for the severity. Protective eyewear may slow down the transmission. I think this is a very important point to elaborate, and I think CDC should, based on the last studies, issue a warning about wearing contact lenses and the importance of protective eyewear. Cyclosporine, there is a big, nice for the study that shows that it can have an antiviral effect against coronaviruses, and cyclosporine, drops are available and they can be a potential option for treating and a prophylaxis for COVID-19. I think, just because it's an urgent matter that we're living in right now, a potential answer for this, hypothesis, and the merit of this hypothesis can be achieved, I think, through a national or a state border survey that looks into how patients that use this, because a lot of people actually use this drop, how these people fared compared to other border people, say in an area where there is high infection rates, say like New York City, how did these people fare in terms of frequency and severity of infection compared to other border people? It'd be interesting to know how they fared with that. Also, those wearing glasses, how they fared compared to those not wearing glasses, specifically in terms of severity of the disease, and also contact lenses, which I assume will be a problem. How did these patients fare compared to people that didn't use it in terms of severity of the disease? That's all what I have. Thank you.
Okay, thank you very much. We have, one question. So the question is, revisiting some of your early slides, is it true that no one has died of COVID in Middle Africa or India?
Actually, this is what I just mentioned in the slide. I mentioned that Ethiopia and South Sudan, which are the countries that have the most, endemic for trachoma, I think one of them, Ethiopia, has zero mortality, literally zero mortality. No one died.
Okay. Are there anybody else like to raise any questions for Dr. Hussein? Fascinating observation. Okay. And okay, and again, if you have any follow-up questions, feel free to reach out to the speakers directly, and we will be querying the speakers later to see if any research projects, collaborative research projects ensued, particularly after this symposium. It's 11 o'clock, and that is Jeroen Pollet from Baylor College of Medicine. And his talk is on immunology and functional assays for SARS-CoV-2 vaccine candidate RBD219. Jeroen, are you there?
I'm here, David. Thank you.
Fantastic. Take it away whenever you're ready. Can you screen share, please?
Yes, I will try to. I can already say I'm also looking at that soluble ACE2. I know ACE2 has a very low half-life in the body.
Yeah.
See my screen?
We see you now.
So my name is Jeroen Pollet. I'm an assistant professor at Baylor College of Medicine, and I work together with Dr. Peter Hotez. I'm basically one of his boots on the ground to make a new vaccine against COVID-19. And we do this at Baylor together in collaboration with PAT, which is the Alliance for Technology and Health. So we're old school. So we don't make an mRNA vaccine. We do not make a DNA vaccine or an adenoviral vaccine. We're making a recombinant receptor binding domain vaccine. And we've done this for over 10 years. We have made successfully vaccines against SARS and MERS, and showed, at least in animal models, that these vaccines can be very protective and safe. And now we want to do the same thing for the new coronavirus. The platform that we're using to make our recombinant protein is a PQ Pastoris platform. And we typically use very simple buffers and have, at least for our previous vaccine, stability for over 36 months. Our formulation is also very old school because we make, so we use alhydrogel, which means that a lot of, actually all of our protection comes from antibodies, which is also very controversial because, of course, you have heard about ADE, antibody-dependent enhancement, but we believe that our recombinant vaccine candidates actually do not cause any ADE. We'll come further into that. We have two vaccine candidates that we are working on. The first one is the CoV RBD219N1, which is the vaccine we made actually in 2013 against SARS. And we have some very good compelling evidence that shows that this could be cross-protective against COVID-19. And I will show you a lot of that data. And then we have also made a new variant specifically for the CoV-2, which is in earlier stage of development. And one of the main reasons that we want to push our original RBD219 to the clinic is because we have GMP material available and we have a lot of safety and tox data on that. So it would be good if we could at least test it in the clinic. So this is some of the evidence that we have against the original SARS with our vaccine. It shows very great antibody titers after immunization of mice. And in the middle graph, you can see that it also has a lot of neutralizing antibodies, actually a lot more if we compare it to other adjuvants that we used. The aluminum came out as one of the better, the ones that had a good ratio of neutralizing antibodies. And on the right, you can see the survival rates. So this is done with an adapted strain of SARS in mice at UTMB. You can see the TBS group, which is a purple group, 6 days post-challenge, they all die. And our vaccine, which is the green one, RBD219N1, has 100% survival. We also have some really good evidence for the safety. So one of the things, of course, is always eosinophilic infiltration, and a lot of the data originally comes from when there's so problems with antibodies is when people are vaccinating with the whole spike protein. As you can see on the right, that also causes a lot of eosinophils. But our vaccine candidate on the aluminum did not show any of that. So we think that because we eliminated some of those immune-enhancing epitopes, we have a better and safer vaccine candidate. And we believe that one of the actually potential mechanisms for immune enhancement is driven by more cellular response, which is Th17 responses. And Dr. Hotes, together with Dr. Corey, actually made a nice publication last month where they linked the Th17 response to IL-6 and IL-8 that is induced through the cytokine storm of people experiencing COVID-19. Also, all our data points out that the immunopathology is actually the least with alum vaccines, which are mainly then Th2 driven, and that is, something that we find against what others are finding. Of course, our vaccine candidate needs to be tested against the new coronavirus, the SARS-CoV-2. Everybody knows that there is a lot of homology between the two RBDs. It's also been pointed out that maybe in the RBM, the receptor binding motif, there are some differences. But it's very important to know that the neutralizing antibodies are not only in the RBM, but also around of it. And a lot of the neutralizing antibodies around the RBM are actually conserved. And you will see in the literature, there's actually quite a few monoclonals out there now that are binding, that are neutralizing and binding to both the RBD of the original SARS and the one of the new coronavirus. We have also tested our serum from mice vaccinated with our RBD 219. So we've used serum from mice and tested it on a pseudovirus platform. In A and C, you can see this is the platform, the original platform we made for the original coronavirus. B and D is a new pseudovirus platform that we developed. So control is this control sera, and then sera from mice that we vaccinated. Our vaccine candidate is in the black bar. The difference between A and C, as well between B and D, is that we used different. We vaccinated with two different candidates. One of them is the top bars are the ones we made in yeast, and the lower ones are ones that we made in a mammalian cell line with an FC tag. This data has been published too. So a summary statement for the first vaccine candidate we have, I think we have some good evidence that this type of vaccine with the aluminum and recombinant protein can lead to 100% protection against coronavirus like SARS. And we have some, I think, good initial data showing that it could be cross-protection with the new coronavirus. And of course, we need to show that now in animal models. But we have some clear evidence for safety, and that would probably go for both the new and old SARS, that the aluminum vaccine and the aluminum vaccines are not as dangerous as some people want you to believe. And because we actually make our proteins in Pichia pastoris, this has been done, for example, for hepatitis B vaccine. So there's a really good track record there with this type of vaccines. A track record which is, by the way, missing, of course, for all the mRNA and the DNA vaccines and the adenoviral vaccines. Finally, just wanted to show you that we are working also on the new RBD vaccine candidate. We are currently optimizing our production process. We have made small batches of different types of the construct. We have vaccinated mice. We have really good titers. They are neutralizing, but it's now a matter of see if we can actually make the yields of the production and the yields of the purification as good as the original one. And then, of course, it's a little bit of a longer way to get to the clinic because we need to set all the protocols, make the GMP products, and do a lot of animal testing before these can progress. I'll stop there.
Thank you very much, Jeroen. Very, very interesting. One question from Christy. Can you comment on the safety of newer approaches that are going straight to human trials?
Well, that's something, yeah, we have, of course, some issues with that. And, you know, it's unprecedented times where you don't need to have your tox study ready before you go into humans. Of course, the mRNA, I mean, there has been some data with other vaccines, mRNA vaccines that have been in phase 1, but we do have some concerns about that. Certainly when we're now gonna start to, if you wanna mass vaccinate, I don't think there is really good safety data out there. That being said, it's a very, I mean, my personal opinion is the mRNA platform is very interesting, and I'm working a lot on mRNA vaccines myself.
Great. Well, thank you very much, Jeroen. There's several other very interesting questions that people have some follow-up on. So, if you don't mind, if you have time, could you join in on the chat box and do your best to answer some of those questions? I'd like to know what those answers are too. So, thank you very much. I'm sorry we have to keep moving on here.
Thank you, David.
Okay, great. So let's move on to Dr. Russell Ray at Baylor College of Medicine, and his presentation will be on mouse models and their models of cardiorespiratory measurement, their rigs and capabilities. Russ, are you there?
We good to go?
Go ahead, take it away.
Thanks. I apologize for the title. I misunderstood things, but I think we got it straightened out, and I appreciate the opportunity to present our work on developing models, particularly towards our interest for understanding SARS-CoV-2 neural invasion and respiratory network disruption, which we think may be a component in COVID-19. I think, as we all appreciate, you know, SARS-CoV-2 infection has impacts that are widespread throughout the body. This is just an illustration from a recent lay review in Science. And the typical path for SARS-CoV-2 to come into the body is through droplets that then find purchase in ACE2-rich cells throughout the upper and lower airway, particularly in type 2 cells in the lung. And when they infect the lung, then what you get, of course, is a neural response and infiltration. You get diffuse alveolar damage. And this all results then in a progression of pneumonia into acute respiratory distress syndrome. And what's going on there is that your lungs are filling with fluid, you're losing compliance, you have reduced volume for gas exchange, and the tissue that can exchange gas has reduced perfusion and of course, cellular breakdown. So that, of course, drives changes in blood oxygen levels and carbon dioxide levels. Your CO2 levels go up, your blood oxygen levels go down, and you have a change in pH. All of that then should be driving neurological responses that are triggered by sensory fibers in your lungs, the carotid and aortic bodies, and of course, complex neural network within your brainstem that senses and integrates all those changes to respond. And those responses typically are a sense of air hunger or dyspnea, tachypnea, which is, of course, an increase in the rate of breathing and depth of breathing. And when things become strongly dysfunctional, then you start sighing and gasping, and you have further respiratory perturbations. Now, what's notable in COVID-19 is that there's large numbers of patients starting or that have been presenting with pneumonia that then rapidly progresses to ARDS. But when they're first examined, they're breathing normally. So they don't show any air hunger or dyspnea. But when you put a pulse oximeter on their finger, they have very low saturation levels. So, you know, some have been reported as low as 50%. They should be convulsing and in seizures at that point. Others are, you know, at 80%, they're talking on their cell phone, and they're completely unaware that their lungs are filled with fluid, and they don't respond to the changes in compliance or the lack of volume. Some people are hypothesizing that, you know, the lack of pulmonary function can result in a reduced oxygen saturation level, but somehow it's enough to still blow off the excess or blow off enough CO2 that you're not triggering these warning centers in the brain. But there's really not good evidence that the CO2 level, what to any changes of CO2 level. I don't think they just have been looked at. So we are hypothesizing that in subsets of patients, SARS-CoV-2 may enter the nervous system through the olfactory bulb, which people have been speaking about. You've heard, I'm sure, all the anecdotal reports about anosmia and ageusia, but or peripheral nerves, say through the pulmonary fibers, to ultimately invade the brainstem cardiorespiratory networks and further exacerbate COVID-19 pneumonia and acute respiratory distress syndrome. And we want to model this in the mouse. And as I was saying, the first question in trying to model this in the mouse is that whether or not SARS-CoV-2 is neurotropic. And there is quite a bit of evidence that it in fact is. Of course, there's the anecdotal reports. There's increasing numbers of case reports of neurological complications with SARS-CoV-2, positive results that, interestingly enough, are otherwise symptom-free. And this is not talking about the silent hypoxemia or silent pneumonia. So there may be other consequences of neural invasion. And there seem to be increasing numbers of retrospective studies that associate neurological manifestations with COVID-19 outcomes. And there's a lot of evidence of other coronaviruses getting into the brain and even causing or likely being associated with neurological sequelae. So we want to try to model this in the mouse. And the reason why we would choose the mouse as a model system compared to a number of other animal models out there is that our facilities are already in place for housing and husbandry. Our lab in particular specializes in engineering mouse strains for functional neural circuit mapping and cardiorespiratory homeostasis and disease. And we are well equipped to measure cardiorespiratory output in the mouse once we have established these models for our genetic engineering. There are 3 models that have been made to study the original SARS virus that in one case has been shown to work with SARS-CoV-2, and will likely the other 2 models work as well. But they are effectively unavailable until this summer. And they're engineered in such a way that you're really overloading the system with high levels of human ACE2 expression so that the progression of infection and disease in those models doesn't necessarily mimic what you might expect in humans. So in order to do that, we're developing these new models based on our circuit mapping approach in collaboration with Koen Venken, I think, who's here in the symposium, and Jason Heaney at Baylor College of Medicine. And the bottom line here is that we engineer synthetic genetic programs into the mouse where we have a ubiquitously expressed promoter whose activity is blocked by these 2 transcriptional stop cassettes. So this is embedded into the mouse chromosome. Each cassette is flanked by either FRT sites or LOXP sites. And these are sites that can be recombined by a site-specific recombinase known as FLIP for FRT sites or CRE for LOXP sites. Many of you in mouse genetics are familiar with these systems. And what's nice about this is we can control the expression of FLIP or CRE recombinase by placing them in front of genes of interest in the mouse that are expressed in specific cardiorespiratory or neural cells, or we can apply them with a virus. In any case, when a cell sequentially, it doesn't have to be concurrent, expresses FLIP and CRE, you'll remove both stop cassettes to allow for the expression of human ACE2 cDNA, thus directing upon inoculation or exposure the virus to infect your cells of choice. So that's seen here schematically, where only cells that have expressed one gene and then the other in the same cell will be expressed with, will express ACE2 and be susceptible to infection in the mouse. Now, to drive that into an ACE2-like fashion, we're of course making a set of recombinases that are targeted to the mouse ACE2. And then each of these recombinases, so we'll make 4 of these mice, can then be further subdivided by your second recombinase of choice that's in the cardiorespiratory system or in the neural system. So for example, this is just again a schematic where we would have the MACE2 flip to remove the first stop cassette, the neurocre to remove the second stop cassette. And only in ACE2 expressing neurons would we express human ACE2 for infection. So this gives us an ability to map in a positive fashion which cells would play a role in various aspects of COVID-19 disease progression.
Hey, Russ, I'm just giving you the 1 minute warning there.
All right, thanks. On the flip side, we're making vectors that allow us to express ACE2 throughout the mouse and then take it away in specific cell types. And we're doing that in a broadly expressed fashion and in an ACE2 expressed fashion here. And the last thing I'll tie off with is that after building these models and challenging them, we have systems in place to take, I think, quite exquisitely sensitive measurements in the mouse in terms of respiratory function, looking at electrocardiograms and blood pressure by wiring these mice up, metabolism, and blood oxygen saturation. So if those are helpful to anybody, please let us know. And these are mice that are conscious and unrestrained. So we don't have to deal with compounds of anesthetics or anything like that. Thanks.
Well, amazing setup, Russell. That's very impressive. So just maybe just one question. Well, actually, there's another question. Are there any, this is from Catherine King, are there any in vitro studies of viral tropism to neurons?
So for SARS-CoV-2, I'm not sure if there's any, but for other coronaviruses, certainly. Most recently, what's been done is there's 2 papers on bioarchives where they've taken involved sea mice, which are more susceptible to SARS and SARS-CoV, is to passage them repeatedly until they become well adapted. So there is a room for initial infection. There are studies with OC43 where they can passage it either through neonate brains, so infect a neonate and then keep moving it along, or through neuroglial cultures to adapt them and then show that they have higher tropism for their brain and greater pathology in the mouse models.
Okay, great, super. Thank you very much. Fascinating presentation. Okay, it's 11:20. Let's move on to Dr. Andrew DiNardo from Baylor. He'll be talking about his randomized double-blind controlled trial evaluating BCG epigenetic and innate training to protect against COVID-19. Hey, Andrew. Perfect. We see you.
Introduction. I'm going to fly through this to stay within my 6 to 7 minutes. So we've known that there's a non-specific cross-protective effect between bacteria since at least 1911. Here you have a bunch of historic studies specific to BCG that have demonstrated BCG's non-specific cross-protective effects. So improving immunity to staph, to other mycobacteria, to listeria, to influenza. To date, studies have shown BCG is cross-protective and induced beneficial immunity to all of these different pathogens that you can see here. So there's also a plethora of data, mostly coming out of Christine Venn and Peter Abbey, starting 1979 up until now, showing that BCG, yellow fever, measles, multiple live attenuated vaccines have off-target, non-specific benefits that improve mortality. So the group of Nijmegen over the past 8 years have shown the epigenetic mechanisms behind non-specific cross-protective immunity. Specifically, they've shown that this is epigenetically mediated and also mediated through cell metabolic changes with an increase in glycolysis inducing the epigenetic changes. So the original paper back in 2012 showed that BCG induced epigenetic mediated changes that improved monocyte immunity to Candida staph. They have many papers on this. Here's one in 2016 that shows that it was mediated through glycolysis. This same first author had produced one that showed benefit in yellow fever. We'll talk about that in a little bit more depth. Maria Lerm had a very interesting paper showing that BCG induces DNA hypomethylation that helps restore and improve immunity. And there was a recent paper showing that BCG vaccinated volunteers that were then challenged with malaria had decreased parasitemia related to the non-specific epigenetic changes. The effects of BCG on viral immunity, there's a review article right here that people can look at later. We're going to talk about the yellow fever study on the next slide, but there was a human study that followed BCG vaccination with influenza vaccination, and it showed that they had improved influenza antibody production as the result of the nonspecific immune boosting from BCG. This is the paper in which humans were vaccinated with BCG and then later challenged with the attenuated yellow fever vaccine. And you can see those that got BCG had lower viremia. And this comes up a lot that people ask about, well, are you afraid if you give people BCG and then they get SARS, they're going to have cytokine storm? But there's not evidence for that. In fact, what was shown here in this study is that all of the cytokines were actually lower except for IL-1 beta. So that most likely what happened is that early innate control of the second infection is decreasing global immunity and this so-called cytokine storm that people are concerned about, and certainly is involved with people that develop symptomatic severe disease due to COVID. So here's all of the beneficial studies that have shown epigenetic mediated benefits. Unfortunately, there's also a lot of detrimental studies that have shown that after severe infection, either sepsis or chronic infection with HIV, chronic infection with schistosomiasis or TB, that you have these persistent detrimental epigenetic marks. And in fact, so we had previously shown that after 6 months after successful deworming, kids with schistosomiasis had persistent detrimental DNA hypermethylation marks. So because of Maria Lerm's previous paper, we were going to implement a BCG revaccination study after deworming these kids. And we were about to launch that before the pandemic shut us down. But because of that, we are working with the group over in Nijmegen. They're a couple weeks, 2 months ahead of us. But here in Houston, LA, Dallas, and College Station, we're planning to vaccinate 1,000 healthcare workers. To enroll 1,000 healthcare workers. They'll be randomized, blinded to either placebo or BCG, then undergo weekly symptom screen, as well as monthly serology and dried blood spot. Whole blood and PBMCs are optional, but to date we've had very good success. Most healthcare workers are very happy to donate a little bit extra blood before vaccinating. And then from this, we'll run through immunology studies and epigenetic studies in order to look at people who develop symptomatic versus asymptomatic disease and individuals, depending on their BCG status, to evaluate the mechanisms by the epigenetic marks by which BCG is hopefully decreasing severity due to COVID. Based on the previous studies, we're hoping to see approximately a 50% efficacy in people that receive BCG. That's it.
Great, thank you, Andrew. That's an impressive study. Is that being coordinated to the TCH network, or are you? Which hospitals are you working through to set up this study?
We have IRB approval at TAMU, College Station in LA, here at Baylor. So any healthcare worker who's interested can either email myself or Sharon Harrison. The urologists were the first ones who jumped on interest in the study because they use BCG every day as first-line therapy, and they know the non-specific immune modulatory effects. So they've been very helpful helping us launch. But any healthcare worker who's interested can contact us. We're trying to target people who are highest risk. So the pulmonologists, the intensivists, the ER physicians, the anesthesia physicians who, no matter what they do, they're quite high risk.
Let me ask you a couple of quick questions also, and from Catherine. How close in time does a BCG inoculation need to occur to provide protection?
Epigenetic benefits are seen within 5 days, but we don't have an answer to that question. In vitro, within 5 days, the epigenetic changes are there. Dr. Rodriguez, we expect a 10% attack rate, and with a 50% with a 50% efficacy, we're hoping to see approximately 20 versus 40 individuals develop symptomatic disease in the BCG arm versus the placebo arm.
Okay. And can you see Andrew's your question from RVS? Could this be why countries with a very high population, such as India, have lower infectivity and mortality rates compared to others? Because they're getting BCG.
It could be. We put a preprint out about that, and we've been hammered on it because of ecologic fallacy. And I don't refer to that as a reason why we're doing the study. There's so many basic science and other epidemiology studies that have shown benefit. But yes, that could be why there are decreased rates. It also could be testing. I mean, we just have to do the prospective trial to figure it out. And we're not thinking that this is the long-term solution. We're hoping some of the other geniuses here can actually figure out how to make a COVID-specific vaccine, and that this just bridges the gap.
Okay, thank you very much, Andrew. Very interesting presentation. There are some additional questions. Again, if we've had a very active and interesting chat going on. If you could hang on and maybe try to address some of the other questions that folks are passing your way. But we're going to move on to Dr. Navin Varadarajan from the University of Houston, who's interested in mapping CTL responses in patients that have recovered from COVID-19 using novel technology. And by the way, if I could just slip in, we have 3 more listed on your agenda, but we're going to try to squeeze in Dr. Rodriguez Baradas at noon because she was unable to make her presentation. So if you can, try to hang on for a few more minutes after noon. Take it away, Navin.
Thank you, David. So if you look at almost all studies characterizing immunity in COVID-19 patients or any viral infectious diseases, we are primarily focused on antibodies and neutralizing antibody responses. And it makes sense because we know how to measure neutralizing antibody responses. One of the biggest headaches with vaccinations and respiratory illnesses, including influenza, which is where I borrowed this slide from, is that it's clear that you need some sort of T-cell protection to ensure that you have lasting immunity against these respiratory viruses. If I'm not mistaken, Andrew, even in the context of BCG, one of the reasons or one of the drawbacks of the BCG vaccine against tuberculosis was that it failed to elicit cross-protective CD8 T-cell responses. And so we as a lab are very interested in this question of how do you map T-cell responses from convalescing patients and how do you use this to make better vaccines. And so if you look at data from influenza and certainly a ton of information from HIV, it's been shown over and over again that polyfunctional CD8 T cells are an important correlative controlling viral replication. And if you look at T cells themselves and ask why are they so difficult to study, unlike neutralizing antibodies, which are soluble proteins that you can map in simple in vitro assays, trying to understand what correlates of T cells are essential for immunity has been traditionally very challenging to overcome. So people study ELISpot responses, for example, looking at secreted cytokines like interferon gamma because it's easy to measure in vitro. But really what the immune response needs to be characterized against is virally infected target cells and the ability to directly map T cells and T cell epitopes that offer CD8 T cells that recognize these infected cells and are able to kill them, and the T cells themselves are able to proliferate. Now, I can argue, and I'm not going to waste your time, that the vast majority of the difficulty here is technological. We have not made very much progress in able to directly map which T cells are able to kill virally infected target cells. And so we developed an assay in the context of HIV infection about 10 years ago. And it's very simple. The images look a little old because I've had to dust it off from the 10-year lag time. But basically, you co-load T cells that you get from either mice or from human patients in this case, with target cells with pools of peptides being presented as antigen by the autologous target cells. So you don't need immortalized cell lines. So you co-incubate both of these in small little compartments similar to ice cube trays, and you monitor T cell mediated cytotoxicity in real time. And then you say, here's a T cell that's capable of killing the target cells that are presenting the cognate epitope. We're able to retrieve this. Once we retrieve it, we're able to do downstream characterization. I'll show you one example we did in the context of HIV. So for those of you who follow HIV, this is a gag-restricted epitope KK10 in the context of HLA-B27 or B57. You had measurable responses in the blood of these patients. And so we looked at interferon gamma secretion or TNF secretion. Typically about 1 to 4% of the tetramers are functional. So we wanted to ask the question, can we directly pick out the the few T cells that are able to kill target cells that are expressing this epitope. And so we did exactly that using the assay that I just described. We have a robot that'll go pick out T cells that are capable of killing the target cells, and then we expand them. After expansion, we show that it is essentially a clonal cell line at this point. All of them stained with the tetramer. Shown here are 3 separate examples of the T clones that we established based on picking directly on cytolytic activity without the need for any other surrogate markers of these T cells. Now, again, we did viral inhibition assays to confirm that the killing function is preserved and that it's able to control viral replication, at least in vitro assays. No surprise, all of the clones that we picked out based on the assay recorded for killing of epitope expressing target cells are able to control viral replication, in vitro at least. And so the second correlative function that I mentioned initially is protein secretion or interferon gamma secretion. It's fairly easy to multiplex this into our assay and figure out how to map multidimensional functional responses of T cells. So in addition to cytotoxicity, we can look for things like interferon gamma secretion. Of course, the big difference is it's all mapped onto the same single cell. So you have cells that are capable of killing target cells expressing the cognate epitope, but they're also able to secrete interferon gamma. We've done quite a bit of work in the context of HIV showing that at least for HIV-specific CD8 T cells, there's no correlation between interferon gamma secretion and cytolytic activity. And so it's a major trend for vaccinology in general on what we want to measure. There's a convenience of measuring ELISpot responses, but if it's not correlated with cytolytic activity or protection, which is very well known in the context of HIV, then really we're just studying whatever is the most convenient to study. And we hope to be able to bridge that technological gap. So in summary, we have an analytical technology for doing integrated analysis of both cytolytic activity and cytokine release. The assay is completely blinded to the epitope. We can just use pools of peptides. So I don't need to know a priori what the epitope being recognized is. We can do downstream epitope mapping studies, and we've done that. And we'd be interested in collaborating with people who are interested in profiling CD8 T cell responses in COVID-19 patients. And of course, with single-cell RNA-seq having made tremendous advances, we can also directly do single-cell RNA-seq on the cytolytic T cells. But mapping, going from TCR to an epitope is still an unsolved problem. So expansion might still be the way to go. But that's my last slide.
Okay. Great. Thank you very much, Navin. Very interesting. Great. It's great technology. We have one question to follow up with. Do you need PBMCs from patients for this?
That would be ideal. Yes.
Yeah. Great. Excellent. So again, thank you very much, Navin. And if anybody has any further questions, feel free to reach out to Navin directly or type in questions here. And Navin, if anybody has any follow-up questions, if you could go ahead and try to address those. It's good, I think, for the entire community to see the answers in real time. And then
I'd be
Suzanne,
happy
could I ask? Okay, great, super, thank you. And Suzanne, we've had a couple of questions about follow-up for seeing the presentation in review. So could you comment on that? Where would that might be available? I understand that we're probably gonna have to make some tweaks to the presentation.
Yeah, so first of all, I'll need to confirm with everybody, we have had some ask not to share it due to unpublished data. One thing that we will have available is the chat so that you can go back and look at the questions and the responses to those. But we will be in touch. I have a list of everybody that's attended, and I'll let you know as soon as we get the final cut edited out, the ones that don't want to be shared, and then we'll make that available on the immunology, the GCC immunology website. But I'll follow that up with an email to y'all.
Great, thank you so much. Okay. It's right at 11:40. Let's go ahead and move on to Numan Oezguen from Baylor, who's interested in computational structural biology, exploring the dynamic and conformational changes of the S-trimer and fusion. So, okay, Numan, we can see your slides. Go ahead and take it away whenever you're ready.
Perfect. Thank you very much, Dr. Corey, for choosing my project for this short presentation. It's about the SARS-2 S trimer and then the cell fusion. So just to be complete, here's the overall structure of the SARS-2 virus, and I'm interested in the S protein and its conformational changes. It has 1273 amino acids. It binds to the ACE2 receptor on the host sites, and on the surface it forms trimers. On the virus surface it forms trimers. So the structure of this spike trimer between SARS-1 and SARS-2 is almost the same. So that's why I'm actually showing here right now the one from SARS-1, just to drive the points. What you see here in gray is the trimer themselves, the spike. In pink are the receptor binding domain on one monomer. And then in green are the ACE2 receptor on the cell surface of the host. So if you go from right to left, you see this RBD domain is in the down conformation over here, while it's here on the up conformation. And at the same time, you see that the ACE2 is binding only to the up conformation of the RBD. So, and at the same time, you see this, oops, sorry. At the same time, what's going on? We see that the binding mode or the orientation of the ACE2 RBD is slightly different in each one of these three structures. So if we go ahead, make this structural superposition, we see that's actually the RBD domain moves as a whole unit together with its ACE2 bound to it. Shortly this year, the structure also of SARS-2 actually came out, and 2 weeks later also the crystal structure of RBD ACE2 of SARS-2 came out. And then if we use now these two structures, what we already know from SARS-1, and make a superposition of the RBD and ACE2 structures, we see that they fit very nicely. And this tells us basically that the ACE2 binding mode in both viruses are exactly the same. So if we look at the sequence of these viruses, we see that the new virus actually acquired a new furin cleaving site at this position here. The red stars here indicate that already it's confirmed experimentally that they are using also furin to cleave the protein. I would like to point your attention here to SARS-1. It doesn't have this furin cleavage site. On the other hand, it's using basically this site here to cleave the whole protein into this S1 and S2 subunits. But this site is also present in the new SARS virus too. Additionally, we have on the S2 subunits in S2 prime cleavage site almost present in all coronaviruses here unlisted. So for fusion, what I see is what we can note is the S protein needs to be primed, i.e. it needs to be cleaved into at least the S1 and S2 subunits, and it needs to bind to the ACE2 receptor. Once this happens, and we don't know exactly which happens first, but once this happens, the S1 components of the whole trimer needs to separate from the S2 so that the S2 core helices are free to change the conformation, their conformations and form the super long helix that will poke later on into the soul and into this host cell membrane. This short movie is just to show you that once the cleavage happens actually at the S1 side with furin, these two parts, this is the S1 part of the trimer and this is the S2 part with the core, they can separate very easily. If anything pulls here on the RBD, pulls on it, it will separate very easily. So the questions that I'm asking now, or what I would like to explore is, first of all, the RBD ACE binding. the RBDase binding, does it happen spontaneously, or does it need any trigger for this to happen? And the second question that I ask is, what are actually the conformational changes that are happening once S1 unit part of the whole spike is separated and not only the S2 part is there present? So what kind of conformational change, how does it start and how is it progressing? This is what I would like to see. So to study this, I applied for large computational resources at the COVID-19 Research Consortium. And I got granted access to the Longhorn computer. This is actually designed for deep learning. That's why they have a lot of nodes and computation power and GPUs. Fortunately, the program that I would like to use, Member 18, is also installed on this machine and also can utilize multiple GPUs. So each GPU has 5,000 processing cores. So if I can utilize, let's say, 2 GPUs, I will immediately utilize about 10,000 processing cores. So this is huge advantage when it comes to the simulations. So before I can start, of course, the simulation, I need to generate the model because the structures that we got for SARS-2 spike are cryo-EM structures, but they have a lot of gaps. So they cover this region from alanine 27 to cerium 1147, but they have a lot of gaps, and I need to fill these gaps. For example, you see a lot of loops that are very loose, and this happens, of course, because they are not structures and they are loose. And then if this is the case, then you usually can see it in the crystal structure or in the cryo-EM structures, and usually they are not reported. So, but if you want to do atomistic detailed simulation, you need, of course, to include them. So fill the gaps, and then add counter ions to achieve electrostatic neutrality. And then finally, I would like to have explicit water around this whole trimer to simulate the system. The system finally, the trimer has 3,300 amino acids. And then if I count together the ions plus the waters and so on, I end up with 500,000 atoms. This is a very large system to simulate. And therefore, actually, the huge computational resources that I granted for to access to, it's actually very welcomed right now. The second question that I would like to ask is, we can generate, of course, this part is by just cleaving the S1 part basically here at this site here, and then we will have the S2 core, and then we can run the MD simulation, just add the waters and condoines and on waters and so on to it, and then we could run it. But I think the cleavage at the S2 prime site here is shown in pink is also very crucial. And the reason why, let me show you why I think it's crucial. So if we see, if you look at this core structure here, when it fuses, this helix will stay put, while this part of the helices need to flip over this way so that it will extend to the a very large helix that can reach then the host cell membrane. But doing so, it will basically pull this helix, it will pull this helix, this will pull this and this one, and then you see this blue one, and here blown up again, you see this blue one here, if it is not cleaved at the pink side, it's actually in front of this helix. It will arrest, it will stop basically the conformational change right there if it is not cleaved. But on the other hand, if it's cleaved over here, this loop regions basically will become very flexible, and they can move away so that this helix here can slide past this coil here and then basically extend this central helix to the final conformation that is capable of entering the host cell. So therefore, I set up 30
seconds.
Oh, yeah, okay. So I do this here, and then the systems are very small, of course, smaller compared to the other one. And I do MD simulations just with two TPU. They progress very fast. So far, the complete trimer, where I try to see whether the RBD movement is going up or no, down, it takes about it progresses 400 nanoseconds till now. The other ones progresses 800 nanoseconds, but up to now, unfortunately, I didn't see the desired movement. And this can have several reasons. One might be that these movements just happen at larger timescales, and the other one, maybe the simulation conditions are not optimal. For example, the pH. This is most probably the most crucial thing that needs to be probably need to change. But first of all, I will run it for the 1 microseconds, and then I will analyze it. And if it's necessary, I'll continue the same thing, or I change the pH, and then I go from there. And I'll stop there, and thank you very much.
Okay, thank you very much. Very interesting, Numan. Due to time, we better stop there. But if you could hang on and ask answer any questions anybody might have, who might be giving you a question there in the chat box. Thank you again. So not quite last. Again, if we can try to get Dr. Rodriguez-Varadarajan after Dr. Koen Venken's presentation. But before then, our next presentation, it will be by Dr. Venken, and who's working with Dr. Andrew Rice. They're generating a platform for rapid construction of SARS-CoV-2 replicons for BSL-2 and BSL-3 level coronavirus research. Dr. Andy, are you presenting, or is it?
I'm going to present some slides first, and then Koen is going to go into it.
Okay, perfect. Thank you.
Okay, so I'm good, and you see the slides?
Yes.
Okay. So what Koen and I our goal is to produce a SARS-CoV-2 replicon system that can be used in BSL-2 containment conditions, which will really facilitate research for a number of investigators. And we also want to develop reverse genetics for the virus, where you can introduce specific mutations in viral genes. And those experiments have to be in BSL-3 containment conditions. And for those of you who aren't aware of that, the BSL-3 the containment conditions are pretty onerous, and the pace of research is something like 1/10 what you would do at the normal lab bench. So so here is the pathogen that we've been talking about. Its genome is 30,000 nucleotides. The first approximate 2/3 of the genome are ORF1A and ORF1B. They encode the non-structural proteins that are involved in replicating the genome. They contain an RNA polymerase, capping and methylation enzymes for the 5' end of the virus is, of course, an RNA genome. So its 5' end is capped and methylated. The virus encodes a couple of proteases that cleave these polyproteins to get the individual subunits. It also contains, interestingly, a proofreading, a protein that gives the virus proofreading ability for its RNA genome, so it can sustain a genome of 30,000 nucleotides, which is the largest of any RNA genome. At the 3' end of the genome are the proteins that are required to bind and enter a cell. The S spike protein that we've heard about, also the 3A envelope and membrane proteins are involved in generating the virus particles. So if one deletes those, you can't produce infectious virus. At the very 3' end are a number of open reading frames that are likely involved in countering innate immunity. My expertise is virology, and Koen's is the construction of synthetic DNA, large genetic elements. To make the reverse genetics, one would clone the entire genome in a bacterial artificial chromosome because of the length of the genome. Then one can transfect this into mammalian cells. In the back, one would have the CMV promoter that allow RNA polymerase to initiate transcription. It would transcribe the 30,000 nucleotides of the genome. There would then be a polyadenylation sequence, in yellow, a termination sequence that would tell RNA polymerase to terminate. And then there's a ribozyme that would then cleave at the poly-A sequence. So one would generate a RNA molecule that is essentially the viral genome, and that would set off infection. And in this system, then one could generate mutations in any of the proteins that one wants, and then study viral infection. But to generate a back, this is taken from a publication with the original SARS-1 virus from the Juanes lab in Spain. And what was done is to delete the spike, envelope, membrane, and 3A sequences and many open reading frames. And then this construct has a CMV promoter to make drive transcription from Pol2, and then the appropriate sequences to terminate and give a poly-A sequence. So this essentially can replicate in transfected cells. It replicates the viral genome. So one can do screens for inhibitors of the enzymes involved in RNA replication. One could also, in this construct, we will introduce a luciferase gene as a reporter to give a very easy way to quantify genome replication. And we're also debating, it would be nice to have a reporter protein such as this strep low affinity nerve growth factor receptor. And what this means is this protein would be expressed at the surface of a transfected cell with a strep region at the surface, and then with biotinylated beads, one could then, in one step, purify transfected cells and do some limited biochemical type of analyses. And one could also add to this replicon various open reading frames that counter innate immunity, and then examine if they have a role in genome replication. Koen is the expert on synthesizing large genomes, so I'll turn it over to him now.
Can I click on these slides or do?
Oh, you'll have to just tell me to advance it and I'll advance
it. Okay, all right, thank you. Yeah, first of all, I definitely would like to apologize because I really got into this about a week ago when I saw a presentation from Andy and colleagues at a Baylor presentation. I've been thinking about synthetic assembly of this virus for a while. You know, it's almost impossible to ignore what's going on globally. But when I saw Andy's presentation last week or Andy's and colleagues' presentation last week, I understood we could contribute to developing one way to synthetically assemble this virus, but also make all sorts of variants. And the reason that I'm saying this is that one, we have experience with working with plasmids up to 200 kb. So that means that actually the coronavirus is reasonably small compared to that. Second of all, we know a thing or two of stitching things together from scratch. So that means we look at things from a Lego perspective. We create blocks, we get them, and then we assemble them at will. And so to give you an example here, without going in too many details, is that we have all this.
We've got one more minute, Koen, just a 1 minute warning there.
Oh, wow, okay. So anyway, we can put these synthetic circuits together. And so we can keep it reasonably simple by going from, a couple of dozen blocks to make a synthetic circuit. And so let's go to the next slide. So currently we are looking at this genome and come up with a way to assemble all of this in a reasonably fast fashion, but also then to make any modification possible for any application. What I would like to promote here is if people really want specific variants or modifications to this virus, being it a non-infectious version or an infectious version, please contact Andy or and me, you know, so we can implement that in the entire pipeline to assemble this virus from starting blocks to a full genome. So let's stop there then.
Oh. Okay, great. Thank you so much. That looks like this is going to be a very powerful technique. And again, you know, Baylor doesn't have much room for BSL-3 work. There may be more in Galveston, but that's tough to work in that environment. So this what you guys are creating is potentially, you know, transformative.
So I want maybe one sentence. Like, I mean, what we definitely will consider is to make it as easy as possible to go from any BSL-2 variant to a BSL-3 variant, to limit the amount of work that needs to be done in a BSL-3 facility.
Terrific, thank you. So if everybody could hang on, we want to see if we can bring back Dr. Rodriguez-Varadarajan for her presentation, which we weren't quite able to get in earlier. Dr. Rodriguez, are you there?
I can see her online, but I don't know if she's hearing us or not.
Okay.
We've made a good effort.
So maybe that won't quite work out.
Okay.
Well, if we'll see what happens here, but I just wanted to again thank all the speakers for really some fantastic presentations. Again, I regret that we couldn't quite squeeze in Dr. Rodriguez-Varadarajan's presentation. Again, please follow up with the speakers, those who are still online. Again, the purpose is trying to generate collaborative work throughout the Texas Medical Center. We're going to be following up with the speakers to see how things have worked out for you in the not too distant future. And I just want to thank again Suzanne and Kara and the Gulf Coast Consortium for helping to put together this excellent presentation. We hope to be doing quite a few more seminars in the not too distant future. Hopefully we can do this in person, but otherwise we'll keep doing these by Zoom.